Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing GATE Biotechnology question paper conducted in the year 2024. I will be discussing the general biology and rDNA questions. Coming to the first question. In adsorption chromatography, the adsorption of uncharged solute molecules onto a silica-based stationary phase is by. So it is by van der Waals forces, or it can be even by hydrogen bonding if the solute molecules have hydrogen bond donors or acceptors. So in the options it is given van der Waals forces, and they have specifically mentioned it is uncharged solute molecules. So naturally the answer is van der Waals forces. All the others, that is covalent bonds, electrostatic interaction, ionic bonds, they are all strong interactions when compared to van der Waals forces, which are relatively weak. Then next one. The relationship that involves the exchange of nutrients between two different species for their mutual growth is called syntrophism. It is also a kind of mutualism. And an example of syntrophism for you to understand easy is that the between fungi and root of trees. Here the fungi helps absorb the nutrients and water, and tree provides carbohydrates. So two different species and there is exchange of nutrients. So it is syntrophism. Now what is antagonism? Antagonism, it is interaction between two organisms where one organism is inhibited or harmed, whereas the other organism is not affected or benefited. That is antagonism. Example is predator-prey relationship. Then commensalism. Commensalism is interaction between two organisms where one organism benefits and the other organism is not harmed nor benefited. Then parasitism is interaction between two organisms where the parasite benefits at the expense of the host organism which is harmed. So here the answer is syntrophism. Mendel's law of segregation applies to the segregation of dash during gamete formation. So Mendel's law of segregation states that each pair of alleles separates from each other during gamete formation, resulting in each gamete receiving only one allele. That is, if you have capital B and small b as alleles, they separate from each other during meiosis. That is, capital B and small b separate from each other during meiosis, and that they are the two alleles of the gene, and each gamete receives only one allele. That is, the sperm will receive either capital B or small b, or the ovum can receive either capital B or small b and not both together. So the answer here is Mendel's law of segregation applies to the segregation of alleles of a gene during gamete formation. Then next one. Co-translational translocation of proteins is observed in. So co-translational translocation, it occurs when the translation of protein and its translocation happen simultaneously, and this is observed in endoplasmic reticulum, and it is seen for proteins like secreted proteins, membrane bound proteins, protein destined to endoplasmic reticulum, Golgi or lysosomes. So what happens here is the ribosome, it will synthesize the protein chain. This growing protein chain, it will be recognized by the signal recognition particle. This signal recognition particle, it will bind to the ribosome and directs the protein chain to the endoplasmic reticulum through the translocon channel in the endoplasmic reticulum membrane. From there, the protein will be transported from the endoplasmic reticulum membrane to the endoplasmic reticulum lumen. So that is how co-translation, translocation of proteins take place. Then next one, 2-mercaptoethanol breaks the dash covalent bond between light and heavy chains of the immunoglobulin molecule. So 2-mercaptoethanol, it is a reducing agent and it cleaves the disulfide bonds between the immunoglobulin light and heavy chains, and it dissociates into components, that is light chain components and heavy chain, and it is used in various biochemical assays, protein purification procedures, etc. So the answer here is disulfide bonds. Answer is option D. Then next one. During normal embryonic development of the mice paw, elimination of cells from the interdigital space is due to. So it is due to the process called as apoptosis or programmed cell death. And this programmed cell death, it helps shape the paw by removing the cells between the developing interdigits. So the answer here is apoptosis. Then next one. A cultured skin fibroblast cell of goat P was fused with enucleated ovum of goat Q. So Q is not supplying nuclear genetic material. The resultant activated early embryo was then transplanted into pseudo-pregnant surrogate female goat R of the same strain as Q. On completion of gestation, female goat S was born. With the exception of mitochondrial DNA, S is a clone of. Now, the cultured skin fibroblast cell of goat P, it was fused with enucleated ovum of goat Q. So P is the only one that is supplying the nuclear DNA, whereas Q is not supplying the nuclear DNA, it is enucleated. But Q supplies the mitochondrial DNA because mitochondria are present in the cytoplasm of the egg cell. So Q is supplier of mitochondrial DNA, not nuclear genetic material. Now R, it is a surrogate mother. So it doesn't contribute any genetic material to the offspring. Therefore, they have asked here in the question, with the exception of mitochondrial DNA, S is a clone of, it is only P. Because Q is supplying only the mitochondrial DNA, not the nuclear DNA, genetic material or nuclear genetic material. So answer here is option A, that is P. Then which one of the following bacteriophages has a genome composed of single-stranded circular DNA? It is X174. Then which one of the following is an insect cell line? It is SF9. SF9 is derived from the ovary of fall armyworm, that is Spodoptera frugiperda. Okay, so SF9 is the insect cell line. HEK293 is human embryonic kidney cell line. DH5 alpha is E. coli bacterial strain, and CHO is Chinese hamster ovary cell line. Then next one. Which of the following is the basic principle of Sanger's DNA sequencing method? Now Sanger's DNA sequencing method, it is also known as dideoxy chain termination method. And in this method, what happens is, once the dideoxy nucleotides are incorporated into the growing DNA strand, it will terminate the reaction there. So you will get different fragments of varying length. These different fragments of varying length are electrophorused on an agarose gel, and from there you determine the sequence of the DNA. So chain termination by incorporation of dideoxynucleotides is the answer. It is not chain elongation. So the answer here is option A. Then next one. An element that is present in a nucleotide but not in a nucleoside is phosphorus. Phosphorus is present in nucleotides, whereas in nucleoside it is not present. It is base and sugar only. Then Krebs cycle is a dash pathway. It is amphibolic. It is called amphibolic because it plays a role in catabolism as well as in anabolism. In catabolism, it breaks down carbohydrate, fats and proteins to produce energy, that is ATP, NADH, FADH2. Whereas in anabolism, it provides intermediates for the synthesis of amino acids, choline and other biomolecules. So it is an amphibolic pathway. The next one. If a denatured protein of human origin is injected into a rabbit, antibodies generated will recognize the dash structure of the protein. So denatured protein of human origin, it is injected into rabbit. Naturally, this will mount an immune response and antibodies will be generated. The antibodies, they will recognize the primary structure of the protein, because the primary structure is unique for each protein and it is not affected by denaturation. So the answer here is option A, that is primary structure. Then next one. All pseudogenes do not code for A. So protein with original function, because pseudogenes are DNA sequences that they resemble functional genes but they are unable to produce the original functional protein because they have mutations and deletions and other kind of disruptions. Sorry. So they do not code for proteins with original function, but they code for RNA with a coding sequence, and this RNA will produce proteins with altered functions and RNA and they can also code for RNA with regulatory functions. So answer is option A. Then next one. If the rate at which E. coli divides is 0.5 per hour, then its doubling time is. Doubling time is also called the generation time, and generation time is reciprocal of specific growth rate. So 1 by 0.5, it is 2. So the answer is 2 hours. Then next one. The decimal reduction time of a microbe during sterilization at 120 degrees Celsius with a first order thermal death rate constant of 1 minute per minute will be dash minutes. Round off to 1 decimal place. So the decimal reduction time, it is the time required to reduce the microbial population by 90% at a given temperature. That is decimal reduction time. And for first order thermal death rate constant of 1 per minute, the decimal reduction time is given by the formula 2.303 by K. So K here is thermal death rate constant, so it is 1. So 2.303 divided by 1, that is 2.303 minutes. They have told to round off to 1 decimal place, so it is 2.3 minutes. Then next one. Match the disease in column 1 with its biological vector in column 2. So Chagas disease biological vector is reduvid bugs. So P4, and that is seen only in option A. So P4 must be, option A must be the answer. We will see. Then trypanosomiasis. Trypanosomiasis tsetse flies. Leishmaniasis is sand flies. Yellow fever is by mosquitoes. So answer A is correct. Then next one. Match the industrial enzyme with its application in column 2. So lipase. Lipase is involved in oil degradation. P2. P2 is seen in B and C. So A and D can be not taken into consideration. So B and C we will see. Fission. Fission is meat tenderization. So Q4. So B will be the answer. R1. Amylase. Amylase is maltose syrup production. That's correct. Glucosidase is oligosaccharide monosaccharide production. So the answer here is option B. Then next one. Match the enzyme in column 1 with its corresponding function in column 2. Now primase. Primase is DNA dependent RNA synthesis. It is involved in the synthesis of RNA from DNA. So P is 4. So P4 means A and D we have to see. B and C we need not take into consideration. Reverse transcriptase is RNA dependent DNA synthesis. So RNA is the acting as the template and DNA is synthesized. So reverse transcriptase is RNA dependent DNA synthesis. RNA replicase. Replicase is RNA dependent RNA synthesis. So RNA is acting as the template and RNA is synthesized from RNA template. So replicase, RNA replicase. DNA polymerase 3. DNA polymerase 3, you all know it is involved in DNA replication. So DNA dependent DNA synthesis. So DNA is the template and from the template DNA is synthesized. So S2. So the answer here is option D, that is P4, Q3, R1, S2. Match the item, that is column 1 with its corresponding use in column 2. Glutamine. Glutamine, it is a source of carbon and nitrogen in animal cell culture media. P3. P3 is there in A and C but not in B and D. So we will see A and C only. Then trypsin is for detachment of adherent cells. Q1. So P3, Q1. Okay, both in A and C you can see P3 and Q1. Hypoxanthine. Hypoxanthine is a component of in hybridoma selection. Hybridoma technology. A component of medium for selection of hybridoma in monoclonal antibody production. R4. So R4 is there only in A1, not in C. So the answer here is option A. Neomycin is for selection of transfected mammalian cell lines. So S2. Answer is P3, Q1, R4, S2. Then next one. Match the chemical in column 1 with its use in column 2. Diethyl pyrocarbonate. It is in prevention of RNA degradation in aqueous environment. P2. So P2 is, you have to see C and D, A and B can be not taken into consideration. Okay, P2. Cesium chloride. Cesium chloride is for density gradient centrifugation, that is separation of biomolecules. You use cesium chloride. So separation of DNA by density gradient centrifugation, Q3. Q3 is there in D, so D will be the answer. Ethidium bromide is for staining RNA in agarose gel, R4. Ethylene diamine tetraacetic acid or EDTA, it prevents chelation of magnesium ion during DNA purification, S1. So the answer is P2, Q3, R4 and S1. Then next one. Match items in column 1 with corresponding technique in column 2. Blue laser. Blue laser is fluorescence activated cell sorting, FACS. P2. So P2, A and B, there is P2. So we have to see which one is correct out of A and B. Tungsten filament. Tungsten filament is electron microscopy. N15 labeled protein, radio labeled protein, is nuclear magnetic resonance spectroscopy, R4. So P2, Q1, R4 is there in option B. So B must be the answer. What about polyacrylamide? Polyacrylamide is used in electrophoresis. That is in page P, polyacrylamide gel electrophoresis. So for protein separation. So a polyacrylamide will be S3. So P2, Q1, R4 and S3. Answer is option B. Then next one. Match the genetic disorder in column 1 with its molecular basis in column 2. So what is the cause of these diseases? Molecular basis. So sickle cell anemia. Sickle cell anemia, it is due to mutation of the beta globin gene, P3. P3 is there in B and C. So we will see which is the right option from B and C. Then xeroderma pigmentosum, it is mutation in nucleotide excision repair. It will cause xeroderma pigmentation. So Q1. So C must be the answer. We will see. Then Tay-Sachs disease. Tay-Sachs disease is mutation in hexosaminidase A gene, R4. And Down syndrome is trisomy of chromosome number 21. So P3, Q1, R4 and S2 is the answer. Then next one. Evolution of wings in bat and insects is an example of dash evolution. So it is an example of convergent evolution, because in convergent evolution, organisms that are not closely related, that is they have evolved from different ancestors, they show similar traits. So here wings of bats and insects, they have evolved from different ancestors. Bat is a mammal, insect is an arthropod. So different ancestors, but they have evolved similar characteristic, that is wings, which help them in flying. So it is an example of convergent evolution. Now what is divergent evolution? Divergent evolution means they have evolved from a common ancestor, but they show different traits. That is divergent evolution. And what is parallel evolution? It is a phenomenon where similar traits evolve independently in different species that are not closely related. That is parallel evolution. And neutral evolution is it states, it is a theory. Neutral evolution is a theory that states that evolutionary changes at molecular level are due to random processes and not due to natural selection. That is neutral evolution. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.